Hi. So now that we've spent some time talking about protein structure, we are now going to discuss the globular proteins myoglobin and hemoglobin as an example of how protein structure affects function. So as you may recall from physiology, hemoglobin and myoglobin are both responsible for the transport of oxygen throughout the body. In this unit, we'll explore how both the tertiary and quaternary structure of these proteins affects their affinity for oxygen and how they work together to help deliver oxygen to our muscles and other various tissues. So through these activities, we will be able to analyze and describe hemoglobin's oxygen binding characteristics using a graph and then correlating the graph with the protein's function. We'll also define and describe the terms cooperative binding and P50 value. We will then describe how conformational changes of hemoglobin caused by the allosteric binding of oxygen is important to hemoglobin's function, and then apply our knowledge about protein structure and function relationships to predict how changes in hemoglobin's primary sequence might lead to pathological abnormalities in hemoglobin function. So let's go ahead and get started with the video.
Hemoglobin and myoglobin both bind and transport oxygen through the body. Hemoglobin transports oxygen through the blood, while myoglobin facilitates the diffusion of oxygen from the blood into muscle cells. Both proteins utilize a heme prosthetic group in order to help transfer oxygen. Where myoglobin is a monomer of 153 amino acids, hemoglobin is a tetramer of 4 myoglobin-like monomers. More specifically, hemoglobin is a dimer of dimers, where 2 alpha-beta pairs come together to form the tetramer. In the center of this conjugated ring system of the porphyrin, the central iron atom is coordinated within the plane by the 4 nitrogens of the pyrrole groups. The remaining 2 coordination sites above and below the iron atom are occupied by oxygen from above and histidine F8 from below. The oxygen molecule is further stabilized by hydrogen bonding with histidine E7 and then locked into place with valine E11 and phenylalanine CD1. Valine and phenylalanine can actually swing in and out as hemoglobin breathes to facilitate oxygen movement on and off the heme. When oxygen is not present, a water molecule takes its place. The binding of oxygen to myoglobin alters the overall tertiary structure of the protein. When oxygen is not present, the central iron atom is drawn out of the plane of the heme through interactions with proximal F8 histidine by approximately 0.05 nanometers. When oxygen binds, that iron atom is drawn back into the plane of the heme, which then pulls histidine F8 with it as well, which then also pulls helix 8, and eventually causes a downstream global shift in the myoglobin structure. While this structural shift in myoglobin is small, we will see a much more profound effect in hemoglobin in both global structure and binding activity after binding oxygen. The binding of oxygen to myoglobin at increasing oxygen pressure or increasing oxygen concentration can be described as hyperbolic, meaning that the binding of one oxygen molecule to one myoglobin does not affect the binding of additional oxygen molecules. Now, this makes sense because there is only one binding site for oxygen on each myoglobin molecule. But let's take the example of hemoglobin, which has 4 binding sites for oxygen. If hemoglobin showed a similar hyperbolic curve, then we would say that the binding of oxygen to the first binding site does not affect the binding of oxygen on the second, third, or fourth site. We will see, however, that this is not the case for hemoglobin, and instead of a hyperbolic curve, hemoglobin has an S-shaped or sigmoidal curve, which we will address later on. But at looking at our curve for myoglobin, we find that the P50 or the pressure at which we have 50% saturation of myoglobin with oxygen, is very low, at 2.8 torr. This means at either arterial blood pressure, which is 100 torr, or venous blood pressure, which is 30 torr, myoglobin should be at least 90% saturated with oxygen, meaning that myoglobin is able to efficiently relay oxygen from the capillaries, high pressure, high oxygen concentration, to muscle cells, which have low pressure or low oxygen concentration.